Hi everyone. It's been an honor to be here. I'm Yong Chen from Bindu University, China. My title is Rescuing KCNT1-Related Deficits in Mutant mice models via AAV Mediated gene replacement. With that, one sentence of context so we are aligned. KCNT1 is a sodium-activated potassium channel. The neurons break. When it's impaired, networks become hyperexcitable, leading to early drug-resistant epilepsy. Now, why these two models? Because they are anchored to real patients. Clinically, we see neonatal infant onset, migrating focal seizures, refractoriness, and developmental impact. This underpins our replacement strategy. Let me introduce the two patients briefly. Patient 1, infant onset, frequent drug-resistant seizures with marked developmental delay. Genetic testing showed a de novo KCNT1 mutation. This case anchors the first mouse model. And here's the second case, patient 2, early infant onset with migrating focal seizures and developmental impairment. Again, de novo KCNT1 mutation. This anchors the second model. With the clinical anchor set, I'll summarize how we intervened. We first construct two-point mutant lines modeling those patients. Having both let us test whether our replacement approach generalizes across background. Here is the therapy and timing. We deliver AAV KCNT1 into the lateral ventricle of 5-week-old mice to drive widespread neuronal expression. After 4 weeks of expression, we run behavior versus mutant and control mice. So let's look at the first model first. On rotarod tests, treated mice stayed on longer. In the pole test, they descend faster. On the treadmill test, endurance is higher. Together, this reflects better coordination and motor output in mutant treated mice. So we saw motor improves, thus cognition follows. In the Y maze, working memory exploration improves, and fear conditioning shows stronger associative learning after AAV treatment. And for spatial learning and memory, we used water maze test. In the Morris water maze, acquisition is faster in treated mice, consistent with improved hippocampal dependent learning and memory. So the first model shows multi-model rescue. Let's see if the second model reproduces this. We see the same model pattern, better rotarod, quicker pole test, and stronger treadmill performance versus model mice after AAV treatment with dose-dependent effects. And for cognition, Y maze and fear conditioning also improve, indicating cognitive rescue in a second genetic background. Finally, spatial learning, water maze performance shifts toward Y-type after AAV KCNT1 treatment, reinforcing generalizability. In conclusion, AAV KCNT1 delivers robust, reproducible behavioral rescue across 2D novel early onset KCNT1 backgrounds improved deficits in rod, pole, treadmill, white maze, field condition, and water maze test. So we are now looking at the EEG, and we think that in 2 months we will see the electrical activity after AAV treatment. So that's our preliminary results. Thank you.